viruses. And in the previous sessions, whatever we have discussed it, a few of you have put some queries and that I tried to reply. So if you had any doubt regarding the general aspect of viruses classification, then please put some questions so that we can discuss in the regular classes. Please, after the end of the class, when you read it once again, please, if any queries or doubt comes, then please contact me and try to clarify those doubts so that you can understand the subject very well. Why we are discussing this way is just for sake of knowledge, to gain the knowledge. So until and unless you participate in this learning process, I feel like your information and the knowledge will not be complete. Anyhow, in our previous class, we talked about a very important aspect of how the virus replication takes place, how because of this unique nature, these organisms, they follow a very peculiar style of replications and totally differ from other living organisms. So there we mentioned about the entire replication stages of viruses into these 5 different categories, like it's attachment or adsorptions. To initiate an infection, the virus must be able to attach to the cell surface, and that attachment, as we mentioned it, that it is through certain specific cell surface receptor. These are certain cell surface proteins which the viruses in the evolutionary process considered as an entry point to the cells. So the virus surface proteins interact with the cell specific surface proteins, and this leads to attachment of the virus on the surface of the cell. And this is essentially needed for initiate and enter and further replications to occur. Okay. So there are certain viruses, as we discussed, like which are very much specific to a particular species of animal. So this is one reason why probably they have the specific receptor for that virus in that particular species of animal. However, there are certain viruses which select some kind of cell surface proteins as a receptor, which are widely prevalent in different life systems, in different species of animal, different even the human. So as a result, those viruses can easily attach and initiate the replications in that particular host. Okay, but not only that attachment, the particular cells where the virus attach and get it adsorbed, the cell must provide the environment for completing its replication. So once the virus get attached, the next stage is the penetration and uncoating. This is simply a review of our previous class. So where we mentioned about that, the penetrations refer to the crossing the plasma membrane of the cell. The virus from outside, they enter inside the cell. And in case of the enveloviruses and in case of different types of viruses, their penetration processes are different. So particularly the enveloviruses, they lift the envelope outside and they come inside the host cells. And even there are certain viruses where the fusions occur. So this penetrations, we refer it to the receptor-mediated endocytosis process, where we mentioned about some specific cell surface protein which act as a receptor for the virus, and that leads to the different method of crossing the membrane. We talk about the macropinocytosis. We talk about the clathrin-coated pits and caveolin protein assisted crossing or penetration of the viruses to inside the host. So once the virus they enter, then next step is the uncoating, where I can see this the nucleic acid structure of the virus get disintegrated, and the nucleic acid releases inside this endosomes, and subsequently it releases to the cytosols. Okay, so this is what we call as the uncoating stage, right? So once the virus get uncoated, virtually we cannot visualize any virus particle. Only the nucleic acid is available till the new virus particle get assembled inside this cell. This is a single infected host cell. So this particular stages we designate it as an eclipse phase of the virus replication. And this eclipse phase is the main stages where the genomic replication takes place, the transcriptions, translation of protein, all the activities takes place. And to carry out this activity, the virus totally depend on the host cell to carry out these activities. So they are, we can say that they totally depend on the host cell machinery for their own synthesis. Since they don't have their organelles to perform this activity, so they depend on the host cells for their replications. Okay, so this is a very unique nature of the viruses. As I told you that if you try to draw the growth of the viruses in terms of the time and concentration of viruses, then we typically get a one-step growth curve of the viruses. Okay, so this three different curve is depicting the cell-free virus, cell-associated virus, and the total virus, like that way. Here we could see that the total viruses, once if you inoculate into an growing system in the cell culture, then after a few periods, the virus number declines. That means this is the eclipse, beginning of the eclipse phase. Then they prepared and all the genomic replication takes place and new virus particle comes out. So this growth curve is designated as one-step growth curve of virus. So this particular period till the new virus particle comes, it's called as the eclipse phase of virus replications, where the virus synthesis takes place. So if you, what we discuss about it, that in this process, once the nucleic acid gets released, then they go for transcription. So the first set of messenger RNA, we call it as the early messenger RNA. And this early messenger RNA carry the genetic information from the virus nucleic acid, and viral protein will be produced in the cytoplasms of the host cell. And these proteins are termed as early proteins. Right? So these early proteins, as we have mentioned, these are basically the enzymes that needed for the genomic replications and transcription. So they utilize these enzymes even to shut down some of the host cell functions so that they can utilize the host cell machinery efficiently. So a lot of roles played by these early proteins. And under the influence of these early proteins, on the second set of messenger RNA or the genomic replication takes place. Taking the original nucleic acid as a template, many copies, thousands of copies of viral nucleic acid takes place. And from those genomic copies, many number of second set of messenger RNA will be produced. We call it as a late messenger RNA by transcription process. And this late messenger RNA again give rise to a second set of protein. We call it as a late proteins. Do you have any difficulty in understanding these stages? Anyone? Could you follow it? Anyone can please reply.
Yes, sir.
Okay, fine. This late messenger RNA will give rise to the second set of proteins. We call them as the late proteins. Early and the late is the time periods only. This late proteins are the structural protein of the virus. Mainly, if it's a structural protein, then it refers to the capsid proteins, it refers to the matrix protein, it refers to the glycoprotein spikes, which are anchored to the envelope in case of the envelope viruses, or maybe the nucleoprotein. So these are the structural protein that will assist or help in formation of new virus. But at the end of this eclipse phase, numerous capsid proteins will be available, many genomic copies will be available. Then this capsid protein will undergo aggregations to form the nucleocapsid structure, and one copy of the viral nucleic acids they will accommodate to give rise to the newly formed viruses. This particular stage is called as the assembly of virion. Okay, so for your information, telling you, but this is just simply keep it as a information. At the later part, when you study more about the viruses, you will be knowing it. This particular process is not a very perfect process. This assembly, what happened when a virus they replicate inside the host cells? Then every time from one virus, when thousands of virus particles will be formed, many virus particles will have defective assembly. We call them as the defective viruses. So those viruses cannot further can cause replication and can cause any disease condition. Only the viruses which could attain the full conformation of the original viruses are responsible for causing further infection in the next cell. So we call them as the defective particles. So maybe in some other classes we talk about more about the defective particles. So the assembly is over. These cells, these viruses will be prepared themselves for coming out of the host cells, and that process is called as the release. Then what we could learn is that the viruses they release by two major methods. One is by budding process. In case of the envelope viruses, whole envelope viruses, they release out of the host cell by budding process. What basically happened, this is an example of the rabies virus from entry to its whole replications. It's showing it here. At the end, when the newly formed virus particle, they will be accommodated at certain small protuberance produced in the cytoplasmic membrane of this host cell. This is the host cell. The cytoplasmic membranes will produce a small protuberance, and through this protuberance, the newly assembled virus particle will accommodate, and finally they will get detached and give rise to an individual virus. So this process is called as the budding. So budding may take place from different parts, maybe from the Golgi apparatus, ear, or maybe cytoplasmic, or maybe nucleus. So it depends on the nature of the virus where they grow and replicate. Okay, whereas viruses like picornaviruses, which are non-envelope viruses, so at the end of its replications, they simply cause lysis of the host cells, and the whole cytoplasmic membrane will get disintegrated, and the virus particle will come out. So that's the last stage of virus replication, the release. This is a very general approach of describing the replication of viruses. Now, depending on the DNA virus, RNA virus, single-stranded, double-stranded, or part single-stranded, part double-stranded, their variations are there. So at this level, we are not going to discuss all the variations about different types of viruses, but rather we will focus it to remember these 5 important stages of virus replication. Okay. So this is another example of coronavirus. See, there are certain viruses which replicate in the nucleus of the host cell. Okay, this is a picture of showing the budding, the scanning electron microscopy of some infected cells where this protuberance produces, and these are the viruses, and finally they will get detached from the host cells. So just the real image of the viruses in the infected cells, and these are the areas where the arrow marks is showing where the budding will takes place, and this like this way the new viral will come out. So I just select these pictures for our understanding. Okay. Now, as I said that the DNA and the RNA viruses in general, the DNA viruses, they prefer to replicate in the nucleus of the host cell. If I ask you a question, why it is so, the simple answer will be that the DNA viruses, they require the host cell enzyme, DNA-dependent DNA-dependent RNA polymerase enzyme for transcription purpose, and that enzyme is available in the nucleus of the host cells. So that is why the DNA viruses, they prefer to grow and multiply in the nucleus of the host cell. Just an example showing it here. So all the DNA viruses, they replicate in the nucleus with some exceptions, like pox viruses. Pox is an example of DNA viruses, but it replicates in the cytoplasm of the host cell. The pox viruses, they carry all their enzymes. They do not depend on the host cell nuclear enzyme for transcriptions. So they can independently grow and multiply in the cytoplasm. These are some exceptions. Similarly, the RNA viruses, they prefer, they do not require the nuclear enzyme system for this replication. We know that there is positive sense and the negative sense RNA. This is an example of positive sense RNA virus where the viral RNA straightway give rise to formation of proteins. Protein synthesis will take place. So they don't require any nuclear enzymes. So that is why the RNA viruses, they prefer to grow in the cytoplasm of the host cells. However, here also there are certain exceptions where some categories of RNA viruses, they partly utilize the nucleus as the stages of replication that we'll discuss in the RNA viruses. Otherwise, the RNA viruses, they replicate in the cytoplasm. Please record it. Now, coming to a very peculiar RNA virus, call it as a retrovirus. Why I am telling it's a very peculiar, because the retroviruses having the diploid genome, their system of replication is different, slightly deviated from the normal method of replication. So here, the virus, it's envelope virus, once they get attached to the host cell surface, they will enter, then penetration uncoating will occur. Once the uncoating will takes place, the viral RNA will convert it into double-stranded DNA using an enzyme called as reverse transcriptase. So it's a unique virus having the reverse transcriptase enzyme in its nucleocapsids. Okay, so using this reverse transcriptase, this enzyme is not available in our body cell. This cell doesn't have this enzyme. So utilizing this enzyme, the viral RNA converts into double-stranded DNA, and thereafter, this is an phenomenal things happen. This virus, they get integrated to the host cell chromosome, and this stage is called as the provirus. Please note it down, provirus. This is a very, very unique characters of the retroviruses. And in the provirus status, the virus genome may remain inside the host for years together. Even if it is a gonadal cell, then it can be transmitted to the next generation. They can pass the information. Slightly just look at the same pictures of retroviruses. So once they enter, then from the RNA molecules, they will be converted into DNA using the reverse transcriptase enzyme, and then they get integrated with the host cell chromosome. Now, these particular cells, when it divide, the progeny cells also will have one copy of the viral DNA. Likewise, it may remain indefinitely within the host. This is the most difficult part to get rid of this provirus status in an infected individual. Probably this is one reason why we cannot get a major breakthrough in treatment of HIV infections. There are many retroviral infections. We are going to discuss it in our class. So in all cases, the major issue is to have effective vaccines against these retroviruses. So this pro-virus status, suddenly at certain circumstances, there are some stimulus, maybe environmental stimulus, hormonal, or certain medicinal stimulus may lead to transcription and translation a new virus particular form, and then they comes out of the host cell. So this is the method how the retrovirus they follow. So these unique things you need to remember. So try to remember the most general aspects of this virus replication. So we'll shift to the next topics of what we call as the inclusion bodies. So I guess all of you have studied many viral infections and you came to know about the inclusions body. But just refreshing your idea about the inclusions body, what basically happens when a virus, they replicate inside a cell. It has been seen that some of the viruses, after virus infection, some remnants means leftover of virus infections, get accumulated inside the cell, and which can be visualized by staining the cells. And these peculiar structures, we call it as an inclusion bodies. The inclusion bodies, why we consider it is important for us is mainly the diagnostic purpose. On the basis of visualizing the inclusions body, these are quite large structure, can be seen on the light microscope very easily after staining the cell, gives us a clue about the virus infection. So inclusions bodies we mainly consider is important for us in terms of diagnosis of disease, okay? But remember, not all viral inclusions are visible. So only where the visible inclusions are forms, we have given specific names and we call them as the inclusions body. But if you try to see what actually this inclusion body, there are many aspects. Like one is the inclusions are the accumulation of viral components. What might be the viral components? It might be the capsids. Many capsid proteins are produced, but some of the capsid proteins are unable to form the nucleocapsid structures. They will get accumulated inside the host cell as an aggregate. We can call it the inclusion body. So some inclusions may be a crystalline aggregates of variants. Some of the viruses, newly formed viruses, because of some reasons, could not come out of the host cells and they get settled at certain compartment of the cells and appear as an inclusion. So inclusions, it's maybe in the nucleus or maybe in the cytoplasm, depending on where this aggregation takes place. Then, not only the viruses, even this is important, like some inclusion bodies are the results of degenerative cellular changes. When a virus replication takes place inside a host cell, obviously the cell injury takes place. Maybe some of the cellular proteins get denatured in the process of virus replications. Viral proteins appear as a toxic for the cellular proteins, and these degenerative cellular changes may appear as an inclusions body. So these are the things where we could see the inclusions body. So for our diagnostic purpose, the inclusions body were classified in different way, like intranuclear, intracytoplasmic, single or multiple, large or small, round or irregular, and then acidophilic or basophilic. If you stain it by hematoxylin and leucine, acidophilic will take the pink and the basophilic will take blue color. So some of the diseases, the inclusions are so specific. For example, in this picture, this is inclusions produced by the pox infections, intracytoplasmic, large inclusions body. You can see these are the normal healthy cells where there is no inclusions, but this area, the infected cells are developing inclusions with an large empty space. These are the large empty space and the inclusions. The nucleus has been pushed somewhere to the periphery of the cell. So this type of pictures, we can immediately say that it might be an pox infection. So as I said, this is important for diagnostic point of view. Similarly, in this picture, can you see the picture?
Yes, sir.
Well, this is an inclusions that's accumulating in the cytoplasm of an infected neuron. This is the negative bodies, as all of you learned about it. And this is frequently infection. This is the nucleus, and this is in the cytoplasmic area where this kind of aggregates takes place. This is such a gold standard. Even now, so even rabies diagnosis, people used to venture to see these negri bodies, and that considered to be a standard test for diagnosis, histopathological diagnosis of rabies. Similarly, so many are there. There are certain infections where both intracytoplasmic and internuclear influences are produced, and some where it is so prominent that looking at these inclusions, we can get the virus infection. This is in a negative body. This is again acidophilic intracytoplasmic inclusions, etc. Okay. Then as I said, the intracytoplasmic or intranuclear inclusions can be formed. Some of the structures is so prominent that looking at it, we can identify the presence of virus infection. Then again, we'll be shifting to the next topic. This is about, obviously, it has come to your mind, like how we can propagate the virus. Well, whenever we talk about the general properties of any organisms, we always discuss about how it can be isolated and how it can be propagated in the laboratory condition. So the system for propagation or isolation of viruses, basically this particular chapter has been kept in your practical, but I feel that it is better to discuss at this point, because when we go for the systematic virology, all the times against the viruses, we'll discuss about this system of propagation. So just like in case of the bacteria, what you come to know that we had certain media, agar media, different type of agar media, which are synthetic media, are available for propagation of the bacteria depending on their nutrient requirements. So proportionately, we mix them, and then components like the beef extract, yeast extract, then peptones, those things we incorporate. And bacteria, happily, they will take the nutrient from the surrounding and they will grow out of their own. But now, all of you come to know that the viruses do not have that particular system of accumulating, assimilating nutrient from the environments and can grow. So they always need a host cell, living host cell, live cells for their growth and replication. Okay, so the viruses cannot be grown like the bacteria in any synthetic media. To make it more simplified, suppose if you just take some nutrient broth or something and put some viruses, this virus are not going to replicate there, because there is no any living cell available in the nutrient broth. So in the process of this development of virology, these are the systems now it is available for propagation of viruses. The living cell system, although it is not in a chronological way, rather it is in the reverse way. The initial stage when people studied the viral diseases, they mainly considered the animal models. They use monkey, dog, then rabbit, mice, where they inoculate the suspensions from infected animals, and they try to reproduce the disease, like the cause postulates. They try to adopt and to see the establish any diagnosis of disease, laboratory animal models. Of course, this is also a system where we can inoculate, the virus will grow and multiply, but this is considered to be a not a superior system for isolating the viruses in pure form. In some instances, we use it. Of course, there are certain limitations again nowadays for using the animal model for isolation of viruses. Okay. Then in our historical development, we talk about this embryonic chicken egg. Woodruff and Goodpasture, these two scientists in 1931 and 33, in between these years, they certainly explored that why we can't use an developing chicken egg for propagation of viruses. And they were very successful at the time and could able to propagate the virus in a chicken egg. A live chicken means an fertile egg, not a table egg. A fertile chicken egg is where inside you will see an embryo, and embryo that grows. When you incubate a chicken egg, it takes 21 days time to hatch the chicks. So the different stages of their embryonic life, we can inoculate the virus material and they will grow inside them. So this is one system. But there are certain limitation in a sense that all viruses cannot be grown in embryonic chicken egg. Fortunately, not only the avian viruses, even certain mammalian viruses can also be grown in the embryonic chicken egg, but that is not for all viruses. There are many mammalian viruses which never will grow inside the embryonic chicken egg. So those are the limitations. So in the gradually, this is an event in 1948, first time this virus, this cells culture system has developed. It is the in vitro propagation of cell. And now we consider it is the fantastic system for propagation of viruses. And if anybody asks you the system for propagation and isolation of virus, always at the top we keep it as cell culture. So for each and every virus nowadays, the cell culture systems are available. Okay, and so even the science has progressed up to such a stage that we can even genetically manipulate some cells to prepare the cell for virus infection. So even some people now also think that probably the SARS-2 virus, because of some of them, this kind of manipulation has get adapted to the human cells. Okay, that's a different issue. So when we talk about this propagation and isolation of viruses, then we always say it is the cell culture system, in vitro propagation of cells. The cells collected from unhealthy individuals can be propagated inside a laboratory, and there we can inoculate the virus and we can isolate the virus. Second is the embryonic chicken egg. So the viruses we can grow in the growing embryo, either duck embryo or chicken embryo, depending on the nature of the virus. And third is the laboratory animal model. Okay, so these are the three systems available. So whenever we'll be discussing about a diagnosis of any viral infections, we'll mention about these systems and what are the changes that takes place in the systems and how we can get the clue that the virus infections taken place. Now, a little bit more information, so I want to share it on the cell culture system. So cell culture, the meaning is the in vitro propagation of cells. That means the very simple way, if you collect some cells from unhealthy individuals, like the blood cells, and then you put it into a flask and give some media, the cells, and provide the same environment that's there inside the animal host, the cells start propagating inside the laboratory flask. And that is what we call as the cell culture. So the cell culture, or more specifically, we designate it as a monolayer cell culture or suspended culture. So first, let us discuss about this monolayer cell culture. Mono means single, layer means one layer of cell culture. Such a thin film of cells, they will grow on the surface of a flask, in a plastic surface, and there the virus infections we can give for isolation of virus. This monolayer cell culture, again, we can categorize as the primary monolayer, secondary monolayer, or continuous cell culture or cell line. These are the three categories. So we'll come to this again one by one. The primary monolayer, the very simple meaning of primary cell culture. From these two pictures, we can get the idea. Suppose you just take some tissues from an mice, maybe an mice embryo, you can take it, or you can sacrifice the mice and take its kidney or its like lymph nodes also you can take it, and then you dissect and cut it into smaller pieces. Initially with some seizure we can dissect it, and after that by using the enzymes we separate the cell from that particular organ. Say suppose the kidney, we collect it from this mice, then using the enzyme we separate the cells, and then we seed them into flax and give some media. So when you incubate this flax, you'll find the cells will start growing inside in the surface of the flax. We call it as a primary cell culture. So primary cell culture refer to growing the cells collected from a healthy individual. So at the time in 1948, it was so popular, and people everywhere in all laboratory conditions they can collect the cells and then they can propagate it. And these systems, when you give the viruses, virus will consider it as a native cells of an individual, and then they will start growing and propagating inside the flax. So you can get the virus in the pure form inside this flask. So this kind of system is called as the primary cell culture. So basically, not only the animal sacrifice, even we can take some embryo. We can sacrifice an animal and kill the embryo, and then we can process it and seed it, and finally we can get the monolayer. From few cells, the cells will start growing inside the flask, and in the laboratory you can get a single thin layer of cells. We call it as a monolayer cell culture. Okay. So at a very regular interval, we perform this monolayer cell culture in our laboratory. Hopefully, when all of you will be returning back, then we can see a session of how we can propagate the cells in the laboratory. Okay, so in some practical sessions, again, we'll discuss more details about this whole systems and the different types of media and the conditions for growing the cells in the laboratory. Okay, so this kind of flax, we just use it to grow the cells, and once you seed it, then these are some they look under the microscopes like the cells. These are the cells which will be propagating, and they will be covering the entire surface. We call it as a monolayer cell culture. Okay, so we call it the primary cell culture when we took the tissue sample from an healthy individuals and process and propagate in the laboratory. Okay. So similarly, depending on the type of tissue you take, it suppose if you take some epithelial, some kidney tissues, then the cells morphology will be somewhat cuboidal like this way. Whereas if you take some fibroblastic cells, then they will be appearing as a spindle shaped. This is an example of chicken embryo fibroblast cell culture. So like this way, we can propagate the cells in the laboratory as a single layer, and once this layer monolayer is available, then we can give virus infections, and the virus will happily grow inside the cells, and they will produce some changes. From few number of viruses, millions of virus particles will be formed. So this way, or this can be considered as a living cell system for propagation of virus, right? So the primary cell culture referred to that kind of cell culture where we take the tissues from a healthy individual. Remember this point. So there are certain issues with this particular primary monolayer, because since we take the cells from a healthy individual, from this particular flask, we can take out some cells and then we can seed it into another flask. We call this a secondary cell culture. We can get the cells there. But this cannot be a continuous process. After several subculture from primary to the secondary, then to the tertiary, like that way, when you go on replicating the cells, after a definite stages, these cells will further not grow inside the flask, because these cells are taken from healthy individuals. So they are not immortal cells. So they will, after a definite period, when the cells will propagate in the laboratory, further, if you try to provide and try to grow them and putting all the nutrients and the conditions in optimum conditions, but still the cells will die off. Okay, so this are the characteristics of this primary cell culture. So to get rid of these issues, people start thinking about some of the cells which can be continuously grown in the laboratory, or some sort of an immortal cells, if you can prepare it. So the first breakthrough has come when this particular story happened. Probably many times you have heard about this name, Henrietta Lacks. So this Henrietta Lacks is a black lady in the US. She developed some uterine tumor, and the doctor attended her tumor, took out some of the cells for diagnosis purpose, and out of curiosity, they just put it into the cell culture flask for growing the cells. So what they observed is that the cells are the cancerous cells, and they are growing very rapidly. So within 24 hours, the cells is doubling its populations. They try to grow the subsequent culture or subculture, and they found that the cells are not dying every time they are going, replicating and forming newer and newer monolayer all the times. And finally, they got it that this particular cells, so which are the tumor cells of Henrietta Lacks, are immortal, and they use this particular cells for propagation of human viruses, and till now these cells are available in our laboratory, and they start giving the name as the Hela cells. Okay, Henrietta Lacks, initials of her name, it is famously known as the Hela cells. So Hela cells are originated in 1951 from this uterine tumor of the Henrietta Lacks. Until now, these cells are available in the laboratory, and we can propagate it. So this gives us a good input as to the people, and they are venturing and try to develop and make some cells continuously growing. So by manipulating the cells, either by physical or chemical method, or even by using some tumorigenic virus infections, nowadays some of the cells can be transformed into a continuous grown characteristics. These cells, they do not die unless you provide the nutrients in optimum level. So this kind of continuous culture of cell are termed as cell line. So whenever you see the word as a cell line, that means these are immortal cells continuously dividing in the laboratory. Okay, so some of these examples like human embryonic kidney 293, then Hela, BHK21, baby hamster kidney 21, then pig kidney, so many, so thousands of this kind of continuous cell lines are available for propagation. So now, as I said, that for each and every viruses, definite cell lines are available, and people are in a pace of research to develop newer and newer cell line for propagation of viruses. Okay, so this all systems what we call as a continuous monolayer cell culture. So these cells, they form a layer of cells. Like this is an again. Viru, it's an African green monkey kidney cell line. Then could you see the cells are propagating? If you take out some cells and put in endoflux and give the growth media, they will grow and multiply, and they will give you a monolayer, single layer cells. And these particular platforms are used for isolation of virus. If you give a virus infection, the virus will attach and they will get absorbed, and then they will start growing inside this cell. Okay. Some more example. This is another Chinese hamster ovary CHO cell line. This is BHK21 baby hamster kidney. These are the cells. Presently, these cells are available in our laboratory. We are propagating it. So these all cells, they are adhering to some surface and they are forming a monolayer, single layer. Beside this, there are certain cells which do not adhere to the surface. We call them as the suspension cultures. They remain in the suspension. So most of the times for production of vaccines, when we need the large amount of cells, then we grow them in the suspension culture, and large amount of vaccines can be prepared. As in the present context, in a bigger, larger type bioreactor machine, the cells are grown in the suspension, and these vaccines are preparing at a higher speed. As you know, there is a huge demand going on, and they are unable to fulfill the requirements, and day and night, these institutes, they are preparing these vaccines for this COVID-19. So basically, they grow the cells in this large bioreactor in the suspension culture. Some other times, again, we'll discuss more about this, just to give you an apprise about the systems. And third is called as the organ culture, where rather than growing the organs, part of the organs are maintained in the cell culture flux, and then we can give the virus infection. This organ culture are basically performed to study the viral pathogenesis rather than in the diagnosis purpose. So when this virus, we give this virus infections to this monolayer cell culture, then we observe some of the cytopatic effect, shortly call it as a CPE, some cytopathogenicity or changes that occur in these cells, like rounding of cells, ballooning of cells, syncytia formations, so where many cells will come together to give the plasma membrane will fuse together to give you an large empty space in the monolayer cell culture. Then inclusions bodies can be formed. Then even there are certain viruses that causes complete degeneration of the cells. So after giving the virus infection, these cellular changes will take place. So here I'm just keeping some photographic image of the changes. This particular picture, refer to this picture. Here we can see this the cytoplasmic membrane get fused together to give it a large empty space. We call it as a syncytia formation. Okay, so the virus is having the definite cytopatic effect. These things we observe after giving the virus infection. Here, this is another picture where we can see the cells get aggregation of the cells, and the other cells get disintegrated. So in the adenoviruses, we could see this kind of aggregation. Here also some cluster formations or aggregation. This is a cytopatic effect. You can see here the disintegration. This was the monolayer. All the cells were intact here. After giving the virus infection, total disintegration takes place. So collectively, we call it as cytopatic effect. So whenever we'll be discussing viral infections, we'll be referring to the specific cytopatic effect produced by the viruses in cell culture, and which is a point for diagnosis. Now coming to the slides once again. So this is the first system, the most efficient system for propagation of the virus, the cell culture system. Then the second system in the embryonated chicken egg. So as you know, like in 1931, the Woodruff and Goodpasture, they developed these particular systems, and they thought of utilizing and growing embryo as an living cell system for propagation of virus. So in fact, the growing embryo, if you see a fertile egg, inside the tissues all are live cells. The embryo, they grow and multiply. They took the nutrition from the yolk, and then gradually they increases its size. So it took almost 21 days time to hatch the chicks out of this embryo. So they thought of utilizing this particular system and the different portions or location of the embryo. If you inoculate the virus material, then specific changes occur. And depending on the viruses, some viruses, they produces very specific changes, and from few number of viruses, millions of virus particles are formed, which helps us in diagnosis of viral infections. So not only diagnosis, even there are many poultry vaccines which are produced by growing the vaccine virus inside the embryo. Okay, so this is the embryonic chicken egg. If you see the different structures inside the egg, then you will find just below the cells, you can get a very thin, fragile membrane. It's called as the chorioallantoic membrane. So this chorioallantoic membrane, the covers and a very large sac inside the egg. We call it as a allantoic cavity. Okay, this is the allantoic cavity. So within this allantoic cavities, you'll find that the embryo is growing in another sac, which is called as the amniotic cavity. This is the amniotic cavity, the spur. So inside the amniotic cavity, embryo grows, and they are directly linked up to the yolk sac, and they are taking the nutrient from the yolk, and they are growing inside this allantoic cavity. So they found that with some route of inoculations where we can deposit the virus outside this membrane, it's called as the chorioallantoic membrane route of inoculation. We can deposit the virus material in the allantoic cavity, it's called as the allantoic route of inoculation, even into the amniotic route of inoculation. Then we can deposit in the yolk sac, depending on the type of viruses they are deposited in this different. And the practical sessions will discuss more about how this procedures can be performed. So these are the different route of inoculation, the different age group of embryo. So like yolk sac route of inoculation, allantoic, then chorioallantoic membrane route of inoculation, amniotic cavity. Even there are certain viruses which are given intravenous injection in the growing embryo. So of large prominent blood vessels spreading into this chorioallantoic membrane, and then with certain procedures we can give intravenous inoculation so that the virus can grow inside this egg. Okay, so in our practical sessions we'll discuss more about the procedure to how this egg can be prepared and can be inoculated for growing the viruses. So when we inoculate the viruses, you can see certain specific changes that takes place inside the embryo. One such is this chorioallantoic membrane, and these are the blood vessels. In the chorioallantoic membrane, it's a very thin layer membrane where certain discrete hyperplastics areas we can see because of the virus infection, and these lesions are called as the pock lesion. So this kind of pock lesions gives us some indication about the growth of viruses. So depending on the type of viruses, different shape, size, and colored pocks are produced in the chorioallantoic membrane. So some of the pocks, they are hemorrhagics, and somewhere it is the discrete white color. So these observations, we do it after virus inoculations, and these are diagnostically important. Besides, we can harvest the antigens from here, and we can detect the presence of virus, growth of the virus from this particular embryo, and then we can detect the virus more specifically by performing the different diagnostic tests for detection of virus. Here in this picture is showing some hemorrhagic lesions on the surface of the embryo. Some embryos are becoming very small. These are some of the changes that are produced in embryonic chicken egg. Okay, so grossly looking at these changes, we can get an idea about the probable growth of the virus. So for further confirmations, we can perform some diagnostic test for detection of antigen inside this growing embryo. So these are the systems that are available for growing and cultivating the viruses. The last system is the laboratory animal. So the specific the animals which are susceptible to the viruses can be used, like the suckling mice. So we can use it, and rabbits, and then guinea pigs as a model. We can use it for propagation of viruses in diagnostic purpose. But this is the least preferred models for diagnosis purpose. For some specific research purpose, of course, people use these animal models, but the most preferred system is the cell culture system, right? So these are the three systems available for propagations and isolation of virus. So in tomorrow we'll have an class in the afternoon half. If you have any queries or any questions or doubts on this particular session, then please do text me, and we can discuss it again in tomorrow about the replications and growth of viruses and propagation of viruses, the system available for propagation of viruses. So this much is for today's session. So tomorrow we'll take a review about it, and then we'll further discuss about the genetic and non-genetic interaction about the viruses, and the host virus interactions that also we'll discuss in tomorrow's class.
Sir.
Yes.
Sir, the positive sense and negative sense RNA viruses, do they play a role in where the virus is to replicate, if it's in the cytoplasm or it's in the nucleus?
No, positive and negative senses doesn't implies whether it will replicate in the nucleus or the cytoplasm. Basically, all the RNA viruses, may be a positive sense or a negative sense, they replicate in the cytoplasm of the infected host cell, with some exceptions where, particularly in the influenza viruses, they carry out one part of their genomic replications. They utilize the nuclear components or some nuclear proteins, and they replicate part in the nucleus. Otherwise, positive and negative sense is not related whether it will replicate in the nucleus of the cytoplasm. So both positive sense as well as negative sense RNA viruses, they replicate in the cytoplasm of the infected host cell. Is it clear to you about the positive and the negative sense?
Yes, sir.
This negative sense RNA viruses, they need which particular enzyme? The negative sense RNA viruses, they require which specific enzyme for transcription? Yes, it is the RNA-dependent RNA polymerase. RNA-dependent RNA polymerase, right? This enzyme is not available in our body cells. So the virus must carry these enzymes in the nucleic acids for initial transcription. But once the initial transcription will takes place, then they can produce this as an early proteins, and again the second set of messenger RNA they can carry it out utilizing their own enzymes. So it's the RNA-dependent RNA polymerase. Okay.
All right, sir. Thank you, sir.
Oh, you want to just give me? Thank you. You might have some other class, I guess. So please do text me some questions. You put it if any doubts are there, and so that we can address in the subsequent class. Thank you very much for attending the class. I'm just closing the link now.